Hello and welcome back to Science of Medicine. Today I want to talk about some basic pharmacology. As you know, in this portion of Science of Medicine, we're talking about some of the chemical applications, and pharmacology is a wonderful application of chemistry as it relates to medicine and integrates with biology and several other fields. And so by pharmacology, what I mean is a very integrated subject whereby the ideas of biology, chemistry, medicine are all combined to study the action of therapeutic agents, or what we would say are drugs, in the body. And this involves a host of people, from people that prescribe the medications to pharmacologists and pharmacists, as well as some of the basic scientists, medicinal chemists, biologists, and toxicologists that basically bring these drugs to market. Now, what I mean by drug is a therapeutic agent that could either be a natural product found in nature or a synthetic molecule that is exerting some beneficial therapeutic effect on the body. Otherwise, I call it a poison if it's a detrimental effect on the body. So this could be something like an antibiotic that will kill things that are invading our bodies. It could be something that is designed to retard the growth of a cancer. It could be something to handle some of the blood pressure problems that many people will experience as they grow older with regard to high blood pressure, or people that have diabetes mellitus, helping regulate their blood glucose levels. Sometimes the drug is really what we call a prodrug, which means that it requires metabolism to be its active form in the body. Now, in the United States, the Food and Drug Administration, which has been around for more than 100 years, is responsible for evaluating new drugs and seeing whether they are actually going to be approved as a therapeutic agent. It does not in any way cover a lot of what are called nutraceuticals or supplemental substances that are sold in health food stores. These do not fall under the purview of the Food and Drug Administration. The process for actually approval of a new drug is very long, many times taking more than a decade, although with the SARS-CoV-2 pandemic, there have been a number of emergency use medications that have been approved. So the FDA ultimately has to answer two questions. First off, is the drug safe or generally safe? It's not to say that any substance that you put into your body is potentially has some sort of side effect. And that's true for anything, even sodium chloride, table salt. What they're generally saying is that on balance, is this generally safe? For example, if you were looking to take a pill for a diet and you took that pill and it caused your heart to stop, that would not be considered to be generally safe, and the side effect of that would not be appropriate for the benefit that you would get for taking it for weight loss. The second thing that the FDA is looking at is if the drug's therapeutic effect is beneficial. In other words, compared to a placebo, is this better? Or in the case of an established therapy, does this drug that is new at least provide the benefit that the current drug provides? So in order to answer these questions, there are clinical trials that are done after the development of the drug, after testing is done on a cellular level and in animal models. And those are based on 4 different phases. The first 3 phases, appropriately called phases 1, 2, and 3, occur before any approval, and phase 4 is post-marketing. In other words, once the drug has been approved and is out on the market, the Food and Drug Administration will collect data related to adverse effects of this drug. So in a phase 1 clinical trial, it's a very small group of people, may only be 10 or so people, and you're trying to answer the question, is the drug safe and what doses are safe? Phase 2, you're also answering questions about safety, but you're trying to understand a little bit about efficacy. Is it working? Is the therapeutic effect there? That also is a small group. Phase 3, if you make it past phases 1 and 2, is designed with a larger group of people to try to test whether this drug actually is therapeutically beneficial. Now, ethically, if I have no treatment, then I can do what's called a clinical trial, where I would randomly assign people to placebo or receive this drug, and then look at the incidence of therapeutic effects. Is it better than the placebo or not? And then also the side effect profile. What side effects were present to a greater extent in the investigational new drug versus the placebo. Now, if I give somebody a sugar pill, there'll be side effects because what will be asked of that patient is to write down every single side effect. So, for example, if they have gas, they'll write that in their logbook. But of course, if you give anybody nothing, or if you give them a sugar pill, we'll have gas with some regularity, or we'll have nausea. And that's why you'll see a lot of these as side effects on typical drugs. That phase 3 trial will then give, along with phase 1 and phase 2, the FDA the information that they need to make a determination of, is the drug generally safe, and does the drug's therapeutic effect exceed that of a placebo. If there is a currently established drug on the market, then it would be unethical to withhold that treatment and give a person a placebo. And so consequently, we compare the new drug with what the established drug is. Most of the drug trials are designed as what are called non-inferiority trials, which means that they're basically the same or not inferior to the current standard treatment for this particular medication. As I mentioned, if that's approved, then we go into the phase 4, which is asking the people that are prescribing the medication to report any adverse effects. And sometimes drugs are taken off of the market as they're tested widely in the population. Now, if I look at a drug, in order to actually have that therapeutic effect, it has to be absorbed, it has to be distributed, and then bind with some receptor that's exerting an effect. And while all of this is going on, I'm also concurrently metabolizing it. So it's a very complicated process, and pharmacologists study this. So in order to absorb the drug, I have to get it into the body. And so the drug or the prodrug is administered to the patient. Now, this could be intravenously or rarely intra-arterially, where I would actually infuse the drug directly. In this case, 100% of it gets into the bloodstream. Most of the time, we administer medications or drugs orally because that's for convenience, and that has to be absorbed in the gastrointestinal tract. Some drugs are readily absorbed and others are not. We'll talk a little bit about a subset of drugs that are acid-base compounds that can be absorbed in various places based on their acidity. Some drugs will then be absorbed to the skin. There are many formulations. For example, Androgel, which is testosterone medication, can be absorbed through the skin. Some contraceptives are actually available in patches. Some drugs can go by way of underneath the tongue. We call this sublingual. Now, in that case, they don't go through the portal vein, which is how the blood is draining from the gastrointestinal tract, and that goes to the liver, where it can be metabolized. We call that first pass metabolism. But sublingual will go directly into the bloodstream, so it avoids that first pass metabolism. Sometimes people are so sick they can't take things orally, and we have a per rectum formulation for the drug. Rarely you will actually put a drug into the cerebral spinal fluid. That's called intrathecal. It's not unusual for several drugs, especially those that deal with pulmonary medications, to have inhalation drugs because they're actually acting on the cells that would line the bronchial tubes. And then sometimes we can administer drugs intramuscularly. Now, once that drug gets into your system, if it's gone through the gastrointestinal tract, as I mentioned, it'll go through the portal vein, which will go through the liver, and there'll be some sort of enzymatic process, which I'll talk about later on. If it's a prodrug, it might be activated to its active drug. Once that drug is in the bloodstream, it will be distributed. So some drugs are highly hydrophobic and some are hydrophilic. If they're hydrophilic, they'll distribute throughout total body water. That means the cells, the interstitium, as well as the plasma. If they're hydrophobic, then they can be put into the fat cells or into the lipophilic portions of the body. Once that distribution is done, then there will be some delivery, of course, because whatever your target enzyme or whatever your target protein or molecule that you're binding with in the drug, it's going to be in one of those compartments that the drug is distributed, and then it will actually bind to that receptor and exert its effect. We'll talk about that. Concurrent with all of this, once you've got the drug in the bloodstream, it will begin to be metabolized. Some things will be metabolized and they will be done through the kidney, then they will be excreted. Some will be metabolized in the liver. Some will actually go through the lung in terms of breathing in and out, such as in anesthetics. Now, ideally, the drug will interact only with its intended target and produce only the desired side effects. But in reality, drugs will bind with several targets, and so that will produce side effects. For example, some chemotherapeutic agents will cause rapidly dividing cells, such as those in cancer, to actually be targeted. But of course, you also have other rapidly dividing cells, for example, your hair follicles, and also cells in your gastrointestinal tract, and those will actually be affected, and you'll have nausea and vomiting or diarrhea. As I mentioned, some drugs will have acid-base properties, and we'll talk about that in a minute, and some drugs can be lipophilic or hydrophilic, thereby going into one of those compartments. While many drugs are small molecules, generally between about 100 and 1,000 grams per mole, many can be larger. That is very true for the monoclonal antibodies and the biologic sections of many drugs that are modulating some of the immunitory response. All right, so let's talk a little bit about more simple drugs that are absorbed through the gastrointestinal tracts and the subset that happens to have an acid or a base moiety, one or more. And so, for example, aspirin, acetylsalicylic acid. Acetylsalicylic acid has a carboxylic acid group. This proton is acidic, and it actually has a pKa of 3.6. Now, what does that mean? So remember that the Henderson-Hasselbalch equation gives us a relationship between pH, pKa, and the log of the amount of base and acid. So if I have an equal amount of in this case, a deprotonated form would be basic, the hydrogen would be gone, and this would be negatively 1 charge, relative to the acidic, which is neutral in this case. And so if I have equal amounts of base and acid, then the pH and the pKa are the same. Well, let's flip that around. If I look at the gastrointestinal tract basically has only a few pHs in it. The most acidic portion is in the stomach, where the parietal cells are dumping hydrochloric acid into the stomach. The pH is about 1. So if I have a molecule such as acetylsalicylic acid, and the pKa of that proton is 3.6, it means that I'm mostly going to have the acidic form of acetylsalicylic acid, and that's the neutral form. Most of the time, the neutral form is absorbed in the gastrointestinal tract. It's easier for it to go across the cell membranes. And so if I look at this in its acidic form, it's mostly going to be absorbed. If I look at things like the ileum and the jejunum, those are on a higher pH, generally around 7 to 8. And in this case, my acetylsalicylic acid is going to be negatively charged. because this proton is going to be gone at a pH that's higher than its pKa. It's going to be deprotonated. And so in that case, it's not going to be absorbed that much down there. So when I look at this and I say, okay, where is it going to be absorbed? As I mentioned, this is mostly going to be in the stomach. And that's for the reasons that I actually then described to you. Let me ask you another one. So this is Itraconazole. Now, Itraconazole is a fungal agent, and it's taken orally. Unfortunately, as you can see from this fairly complicated structure, this is not exactly the most hydrophilic type of molecule. It is actually very, very water insoluble. And so basically, in order to get this solubilized, it has to be protonated. And you can see here in terms of this species is overall positively charged when there's an extra hydrogen on one of the nitrogens. Now, its pKa is 3.7. So in order for this substance to be even soluble and not be insoluble in your GI tract, you need to have it protonated. This is one of the few examples where you need a charged species in order to get across the membrane because it's just not soluble otherwise. So in this case, what part of the gastrointestinal tract do you think that this would be soluble? And then a second question is, patients that are taking itraconazole are generally advised to avoid proton pump inhibitors such as Nexium that block the release of acid into the stomach. Why is that so? Well, the first thing that I hope that you would see is that in order for this to be protonated, your pH has to be lower than 3.7. And the only portion of the GI tract where that's the case is the stomach, which is a pH of 1. And so this is mostly going to be absorbed in the stomach. The reason for reducing the Nexium or eliminating the Nexium when you're taking itraconazole is because, well, if you block the hydrofluoric acid proton pumps from the parietal cells, then the pH won't be as low and you won't have as much of the protonated form of the itraconazole and it won't be absorbed. In fact, sometimes physicians will actually even recommend that a person drink an acidic compound such as a juice or even a soda when they take this, just to lower the pH a little bit more to make this more soluble. All right, good. So let's talk a little bit about bioavailability. Now, as I mentioned to you before, I can give a drug many different ways. If I give a drug IV and I plot the concentration as a function of time and I measure the concentration of the drug in the blood, if I give that drug IV, immediately as I give that, that dose is going to be administered and going to be in the blood. So 100% of the drug that makes it out of whatever bag or whatever that I'm actually giving the drug in through the vein, 100% is going to be in there. So if I gave 100 milligrams, I'm going to have 100 milligrams in there. It will go down because, of course, I'm actually having it being eliminated, which I'll talk about a little bit later. And by virtue of this, the bioavailability of an IV formulation is by definition 100%. On the other hand, if I give it in an oral form or per rectum or something, and I measure the concentration in the blood, the rise in concentration is slower because it's not instantaneously getting from your stomach or gastrointestinal tract immediately into the bloodstream. I also have first-pass metabolism, which lowers that concentration. And so I get this more peak-shaped or rounded kind of curve. Now, if I look at the amount of drug that I've given, and I look at the amount of drug that I actually get in the system, I can look at that and say, oh, I only get 50% of the drug. My bioavailability is only 50%. And that has important ramifications for dosing in terms of how much you would actually want the person to take. One thing is that if you have a medication and then you have generic medications, one of the things that have to be shown by the generics is that the area under the curve, this blue portion, for example, has to show the same bioavailability. And this is one of the things that is done after a drug is off of its patent protection. So once my drug is actually in the appropriate compartment, and it can interact with whatever it's trying to bind to. And pharmacologists define two different characteristic kinds of names for drugs in terms of what they do. So an agonist is anything that will bind to a receptor that causes activation of some process. And remember that we talked about some time ago various cellular pathways. So for example, a drug could be could bind to, let's say, an enzyme link receptor that would then cause activation of some process. An antagonist will bind to a receptor and it prevents the binding of another molecule that would activate the receptor. I'll talk a little bit about some of the slight variations in terms of these terminologies, but agonist is a drug that's going to bind to the receptor causing activation, and antagonist is going to bind to the receptor but prevent the binding of another molecule. And these receptors can be up-regulated or down-regulated on the cells based upon various things. Now, quantitatively, if I look at those receptor-drug interactions, I could look at a dose-response curve. So response versus a dose. And this is generally plotted as a semi-logarithmic plot where the concentration or the dose of this, in this case an agonist, something that's activating it, will actually toggle across many decades. And it will follow this sigmoidal type behavior. That's very, very common. And so if I quantitatively describe this drug effect where I have some sort of maximal effect and concentration, I find a couple things. First off, this is very reminiscent, for those of you who have taken biochemistry, of a Michaelis-Menten kinetics type expression. Now, the one thing that I will point out here is what's called the EC50. The EC50 is a drug that produces 50% of the maximal effect. So in this case, this drug went from, let's say, maybe about 20% up to about 100% effect, and the EC50 would be then at the midpoint between that and down at this dose, in this case 1 molar. So this type of sigmoidal dose response curve is very typical, and that's because of the kinetics that actually describe this. When I'm looking at these dose response curves, there are two characteristics from these curves that I want to elucidate further. The first is called the efficacy. The efficacy refers to the effect the drug exhibits at a given concentration. So for example, if I look at this blue curve that's here versus this red curve, at a concentration of, let's say, about 10 to the minus 6 moles per liter, at about 10 to the minus 6 moles per liter, the effect is the same for drug A and B, for the blue and for the red curve. But if I go to a higher concentration, let's say 10 to the minus 5th moles per liter, then the effect of the drug that's blue, represented here, is higher than the one in red. So I could say that at a concentration of 10 to the minus 5th molar, that drug A has a greater efficacy than drug B. Likewise, if I were at 10 to the minus 8 moles per liter, the blue curve hasn't even begun to rise up, whereas the red curve has, and so the efficacy at that concentration is greater for the red drug, or drug B. Now, if I look at the maximal efficacy for either drug, you can see that the blue drug is actually more efficacious than the red one at maximum, because at any concentration. The second thing is the potency. The potency refers to the concentration of the drug that's needed to produce a desired effect. Many times, we do this at the 50% mark. So for example, looking at this drug B, which is represented by this red curve, at 50% of the maximal effect for it, the concentration is about 10 to the minus 8 moles per liter. And similarly, at about 10 to the minus 6 moles per liter, the potency of the drug here, that potency of 10 to the minus 6 molar for the blue drug has an efficacy of about 50% of the maximum amount. So if I compare the potency of these, the drug B, which is the red drug, is more potent. Now the reason that I distinguish these two concepts is that efficacy is really determined by that strength of interaction between a drug and its receptor. In other words, when the drug actually will bind with the receptor, how much of an effect does it exert? On the other hand, potency has a lot of things that determine it. The drug has to make it to the receptor, which has to do with its metabolism, its distribution, its absorption. And so they're really looking at different kinds of things. But they're important concepts. Let me make sure you have those down. That's right. The curve that is represented by A has the highest potency. The concentration necessary to get its half efficacy is something less than 10 to the minus 9th moles per liter, compared with, let's say, the drug B, which would be maybe about 10 to the minus 8. On the other hand, the drug that's represented by the curve C will have the greatest maximal efficacy. In this case, since it's measuring stroke volume increase, it would actually go up to about 80%, whereas A, B, and C are less than that. Good job. Now, I said that there were some slight variations in terms of definitions. I'm going to go back to my agonist and antagonist idea. So the agonist binds and causes activation. But it turns out that drugs will bind and they will activate, but to different extents. So I use the term full agonist where I get the maximum effect. Partial agonist where I don't get the maximal effect, but I get some effect. Sometimes we use terms like inverse agonist where I actually get a negative effect. I'm not going to talk about that in terms of this. So partial and full agonists are useful, and we'll talk a little bit about that. Now, I'm going to give you the pictorial version of this. If I look at an agonist, the agonist is binding to some receptor, and I'm getting activation. In this case, it would be, let's say, a full agonist, and I get 100% activation here. Likewise, if I had an antagonist, it could bind with the receptor and then block the agonist from binding, so there's no activation when I have the antagonist. What about if I have both the agonist and the antagonist together? Well, I'm going to have less activation because I'm going to have some time where that antagonist is actually blocking the agonist, and maybe sometimes where it's not. And it depends, what that looks like depends upon the nature of the antagonist. And we'll talk about that in a second. If the agonist and the antagonist are competing with each other, that is, that the antagonist is a reversible antagonist. It will bind in the receptor site and then unbind and go through a competition with the agonist. Then what I do is the shape of the curve, that is the dose-response curve, is the same. I still get the same maximal efficacy, but it is shifted based on the antagonist's concentration. So as I have higher and higher concentration of antagonist, it lowers the potency of that drug, of that agonist. And so I'm showing here in these circles where I have just the agonist, and then in these diamonds over here, I have the highest concentration of antagonist, and it's shifting the potency. In contrast, if I have an irreversible antagonist, that antagonist will bind with the receptor and deactivate it. Most of the time, these are chemically modified reactions that make it so that the agonist can no longer bind. And when that happens, the maximal efficacy is changing. If I have the agonist binding, my dose-response curve would look like this. But if I have an irreversible antagonist, I will lower the efficacy depending upon the concentration of the irreversible antagonist. And you go, why would you want to do something like this? Well, you may or may not want to do something like this, but for example, chemical warfare agents will do this. Certain pesticides will do this. They'll bind with that receptor site, and they'll essentially knock it out so that the agonist cannot bind. This also is what it looks like if I deplete receptors, because if I irreversibly bind to the receptor, it's as if it wasn't there to begin with. And sometimes receptors will be upregulated or downregulated. So if you downregulated receptors, that is, you decrease their amount on the cell surface, then it looks like an irreversible antagonist because the maximal efficacy changes. All right, I mentioned to you about clearance of drugs. And so once a drug is in your systemic circulation, your body understands that this is not something which is normally supposed to be in your body. We didn't grow up evolutionary-wise on ibuprofen or Percocet or some antibiotic that we use. And so those are foreign substances to the body, and the body will try to eliminate them. There are several ways that it can do that. If the drug is inhaled, it can simply be exhaled. And a number of inhalation drugs are simply cleared that way. For example, anesthetics that are breathed are cleared by exhalation. If the drug is water soluble, then it can be cleared by the kidney, because if it's a small molecule, the glomerulus is going to allow that to go directly into the filtrate. And the proximal convoluted tubule is not going to have a receptor to reabsorb that drug. And so it's just simply going to be cleared renally. But sometimes drugs are not that water soluble initially, and so those are going to have to be cleared through the liver. And what happens is the liver has two general classes of making those molecules that are drugs able to be eliminated. That process by which those hepatocytes clear that drug or make it so that it can be cleared is called hepatic clearance, and it's actually occurring via what's called the cytochrome P450 enzyme pathway. There are others that are also cleared in that capacity too. Now, phase 1 reaction are going to convert that drug into a more polar metabolite by adding some group, like a hydroxide or an NH group, to make it so that it's more water soluble, so that, for example, it can be cleared in the kidneys, it can be excreted. Sometimes that won't do it, and you need to go through phase 2 reactions. Phase 2 reactions are where the hepatocytes will couple the drug to something else. Sometimes it will couple it to something else which can be eliminated, let's say, in the gastrointestinal tract, or when it make it coupled to something that will allow it to be cleared through some other mechanism. But one of the things that I want to talk about with regard to hepatic clearance is very important because it has to do as the basis for many drug-drug interactions. So the modifications in the liver by the hepatocytes occur via this cytochrome P450 pathway. And that pathway involves enzymatic changes, adding hydroxyl group, coupling, or things like that. And those enzymes can be either ramped up or ramped down. And that is the basis for a number of drug-drug interactions. And what can happen is you will be taking a certain drug, and then you add another drug, and it doesn't interact directly with the drug that you're already taking. But what it does is it causes either the enzymes in the same pathway that are metabolizing the first drug to be upregulated or ramped up, or ramped down, slowed down. So we call those different classes of medications or classes of things that will actually accelerate or decelerate the enzymatic reactions in hepatic clearance. We call them either inducers, which would be to make it more likely or faster for those enzymes to actually clear, or inhibitors, where it would actually decrease the rate of these phase 1 and phase 2 reactions. And this happens all the time. You always have to consider this if you're a clinician and you're actually prescribing drugs. So for example, one of the typical things that will happen is a patient will be hospitalized, and let's say they have a pneumonia, and you do IV antibiotic medications for that. But those IV antibiotic medications might be metabolized via the same pathway and might cause inducing or inhibition of those cytochrome enzymes. And let's say the person that has the pneumonia also has a seizure disorder. That is, they have abnormal electrical activity in the brain, and they're on medications that are called anti-epileptic drugs. Those are drugs that are there designed specifically to tamp down that abnormal electrical activity so that the person doesn't have a seizure. Well, many antibiotics will actually cause those anti-epileptic drugs to be metabolized more rapidly. And when that happens, the person will come in, you'll give them antibiotics, and then they'll have a seizure, because that is all those anti-epileptic medications, their concentrations have been altered by this pathway being ramped up. And so you're actually not able to protect the person against a seizure. So these are very, very important considerations. They come up as drug-drug interactions, and so now there is software to help clinicians actually modify things. As I mentioned, on the kidneys, if it's water-soluble, it's a small molecule, it will generally be excreted, and in the lung, again, it can be exhaled. So how does this actually occur on a quantitative basis? Now, most medications are eliminated via first-order kinetic process. That is, the rate of diminution in that drug concentration with time is equal to a rate constant times the concentration of the drug, first order. And if you separate variables and integrate that, you'll see that the concentration of the drug at any one time is equal to the concentration of the drug that you start with times the exponent of the minus the rate constant times the time. And so it follows, if I look at the concentration of the drug this time, this exponential decay. And many times it's useful for me to define what's called a half-life, which is where half of the initial drug concentration is present, and I can calculate from that half-life a rate constant that allows me to do this. And that's useful. I'm gonna talk about that a little bit more when I talk about dosing. The one exception that I will mention to this is ethanol is eliminated in a zero-order reaction, which means that the concentration that is present in the blood is linearly decreasing. It does not depend upon the amount of drug that you actually, or ethanol, you actually consume. So once you consume a certain amount of ethanol, it will take a certain amount of time to eliminate that. And it's not dependent upon if you imbibe double that concentration, it'll take double the amount of time to actually eliminate that. And that's not true for first order processes. So this is important. This is important because when we dose drugs, we will generally do multiple doses. If you have an infection and you're given an antibiotic for, let's say, a urinary tract infection, you might have a 5-day course or a 7-day course if you're a guy. And so you'll take that drug once a day or twice a day. And it doesn't automatically just go up to some concentration and stay there. It increases in concentration and then it's eliminated. So I've tried to show with this graph, if I look at the concentration of the drug on the y-axis with regard to time, I give a dose and then it starts to decay, and then I give a second dose and it starts to decay, and I give a third dose, it starts to decay, fourth dose, fifth dose, sixth dose, so on and so forth. You can see that after about 4 to 5 dosing, if I dose this at an appropriate time, that I'll approach what I call a steady state concentration, kind of an average between the low point, which we call a trough concentration, and the high point, which I call a peak concentration. And initially, it takes a few half-lives for that drug to reach that steady state concentration. And if you think about it, you dose it at this half-life. After 1, then I get 50% of that steady state concentration. After 2, I get 75%, 3, 87.5, because I'm just dosing and adding half of the life. So half of 100%, half of the 50%, half of the 25%, half of the 12.5%, and so on until I get towards this steady state concentration. And ideally, what I want is the steady state concentration to be sufficiently big that I will have my therapeutic effect. Now, sometimes we will have what's called a loading dose, which means that I'll give a very large dose initially. Remember that volume of distribution that I mentioned to you before. I might be able to calculate from that volume of distribution how much I need, and I'm going to give a very large loading dose to sort of approach this steady state from above as opposed to from below. And that's for very serious kinds of things that we want to do. For example, in the case of a person with a seizure disorder who's actively having seizures, I want to give a loading dose because I don't want that person to seize and die. Now, ideally, what I have, if I look at this steady state concentration here, is that I have some minimal therapeutic concentration, and this should be certainly at the minimum at this steady state trough, and I have some toxic concentration, which I'm hoping is well above the peak concentration. And this range of concentrations from the minimum therapeutic concentration up to the toxic level is what I call the therapeutic range. And that therapeutic range hopefully is going to be larger than these variations in my dosing schedule. Some drugs you have a very narrow therapeutic range, and other drugs you have a fairly wide one. So how do I actually figure out my dosing? So when we're dosing drugs, I normally am trying to dose with things such as trying to achieve a certain dosing rate. And I need to figure out how much drug in order to do that I need. So it's a pretty simple calculation. I'm going to use my dosing rate and my dosing interval to calculate that maintenance dose. But I have to remember that my bioavailability is in here. If I have an intravenous amount, then that's 100%. Bioavailability is 1, and it's no big deal. So if I have something like vancomycin, which has 100% of bioavailability if I give it IV or intravenously, if I have a dosing rate of 80 milligrams per hour and I'm giving it every 12 hours, that means that I will need to give 960 milligrams of vancomycin for that maintenance dose. If I had some drug that had a bioavailability less than 100%, like let's say it were 50%, then the bioavailability would be 0.5, and the maintenance dose would be double that amount. I hope this has given you a little bit of a taste of pharmacology, a very, very wonderful study of how chemistry is used in medicine. I also wanted to let you know that this will bring to an end for this semester the Science of Medicine class. And I've noticed this was originally intended for students that were taking my Science of Medicine class, but I've noticed that a number of people have been watching this outside of that class, which is great. I'd like it if you would leave me some comments about this, because I may actually go and add to this quite substantially in the next semester when I do a medical physiology class. So if you're one of those people that are there, have been watching this and enjoy it, leave me some comments, subscribe so that I know who actually is taking this and if there's interest in doing this. I have gotten some feedback from some of my students that will want to make these segments a little bit shorter, and so I'll try to do this in 30-minute intervals if I do this for the medical physiology. Anyway, I've enjoyed being with you in Science of Medicine. We may see each other again, and give me some comments and subscribe. Thanks a lot.